Point mutations by Vanch. Syllabus identification. Compare the causes, processes, and effects of different types of mutations, including but not limited to point mutations and chromosomal mutations. Terms and pre-presentation knowledge. These are useful for gaining a better understanding of the process which occurs during point mutations and how it occurs. These are also extremely crucial to understand as they are the key terminology which is used inside of the scientific point mutation report. Codon, a sequence of 3 consecutive nucleotides in a DNA or RNA molecule that codes for a specific amino acid. Amino acids. The building blocks of proteins are amino acids. You need 20 of these molecules for your body to operate correctly, since they are the building blocks of life that are required by all living organisms. 11 of the essential amino acids are produced inside of your body. A polypeptide is a continuous unbranched chain of amino acids, joined by peptide bonds. Moving on to mutagens. Mutagens can cause mutations to occur. The first type of mutagen is electromagnetic radiation. Electromagnetic radiation with high energy can cause cells to vibrate, which then lose atoms. These can occur through UV lights and X-rays. Chemical cause mutations by making gaps in DNA, incorporating themselves in DNA, and inserting themselves into DNA. Naturally occurring mutagens. Naturally occurring mutagens come from plants, animals, and microbes. Introduction. What are point mutations? During the complex process of DNA or RNA replication, mistakes can sometimes occur. These mistakes, in scientific terms, are known as mutations. Mutations are when a change is made to the DNA. Types of point mutations. Types of point mutations. There are 3 different types of point mutations: substitution, deletion, and insertion. Substitution. Substitution mutations are when a base pair inside of the DNA is substituted for a different base pair. There are 3 types of substitution mutations: missense, nonsense, and silent. Deletion mutations are when a base pair is removed from the triplet base sequence, also known as frameshift mutations. Insertion mutations are when an unnecessary base pair is added to the triplet base sequence, also known as frameshift mutations. Substitution, silent. Silent mutations can be seen here on the model. Silent mutations occur when a single nucleotide is changed to a different nucleotide. However, the set of the 3 bases still codes for the same amino acid, and thus the same polypeptide chain. This can be seen by G being substituted for C, though the result of the amino acid still remains the same. Moving on to substitution mutations, nonsense. Nonsense mutations can be seen here. Nonsense mutations occur when nucleotides are substituted, leading to the formation and creation of the stop codon. Here, this can be seen as C has been substituted for A, resulting in the stop codon. This stop codon will lead to prematurely stopping the growth of a polypeptide. Moving on to substitution, missense mutations. Missense mutations can be seen here on the model. Missense mutations occur when the nucleotide substituted leads to the coding formation of a different amino acid. This can be seen here as G has been substituted for C, which results in a completely different amino acid. The next type of mutation is insertion, frameshift. Insertion mutations can be seen in the model here. Insertion mutations occur when an unnecessary base pair is added to the triplet base sequence. This can be seen here, where C is added to the multiple triplet base sequences, rendering the rest of the chain useless. Deletion, frameshift. A deletion mutation can be seen here at the bottom of the model. Deletion mutations only occur when a base pair is deleted from the triplet base sequence. This can be seen here. When A gets deleted, the chain moves in together. This stops the growth of the polypeptide, similar to insertion. Frameshift mutations overall. Insertion and deletion of a nucleotide is often studied together, due to both of them having an extremely similar impact on DNA. Both mutations strongly impact the sequence of amino acids, as can be seen here. Which will be produced if one or two different bases are added to the 3 base codons. It can alter the chain preceding the mutation. Nucleotide deletion or insertion causes the grouping to alter and modifies the coding of all downstream amino acids. Typically, the protein that is generated as a result is not functional. Thank you for listening. This is the point mutation presentation by Vanch, and it ends now. Alright, so some extra information. So this I used here is magnetic paper like this, because even if I do this to the board, it's not gonna fall, it just stays on. I've used these tiny magnets to secure it in place, just in case, like wind, me bringing the model to school, whatever. And by the time of marking, I'm not gonna be here because I'm going to India, but the magnetic paper might actually not work properly, like it might slowly be falling off, so I recommend just using the board flat on the ground, like this, on any flat surface, and then just moving around the different nucleotides, codons, and then experimenting with the thing. And so these labels are exactly what is on the slides. So deletion, exactly same thing on the slide. I know we're not supposed to write too much, but I just did this just in case, because without a presenter this would be much more useful. So if there wasn't anybody presenting it, then they would be able to look at the labels. That's all I wanted to say. Thank you.